So thank you very much for inviting me to come and give a talk about what we are doing. I'm the odd man here because it's home, but far away from our home, about 500 miles above us. So this is one of the best classical example of human-built environment. So today I'm going to talk about the environmental omics of space station. So I'm coming from a group called Biotechnology and Planetary Protection Group. So we are chartered to protect other planets. So while we are doing that, our main work is do not contaminate the exploring planets like Mars. So for that we develop a lot of cleaning and sterilization technology. So those technologies are being used for human presence in other planet. So why NASA care about microbiome? So as I said, human habitation in Mars and beyond is one of the biggest thing. So if you are having the human living together and then breathe what they are, exhaling, is one of the things that you have to be very careful. And unlike the built system here, you cannot exchange the air from outside. So you have to revitalize your own air. So that is the biggest challenge. So that means, sometimes the microbes that withstand such an air handling system will become a robust bug. So that might be a huge impact on the human. So as a built-in environment, we need to get the information about what we are living in. So that will give us some sort of sense about what appropriate countermeasures needs to be developed. So this is one of the things that we have to think about. At the same time, we have to utilize the space for producing some novel drugs. So intentionally we will explore the microbes under microgravity to see whether that particular microgravity stress is inducing these bugs to produce some sort of secondary metabolites that can be useful. So, and also the space radiation is another issue that these microbes might cope up and then survive. So I will touch these two things. The majority of my talk will be about the built-in environment. So what are the closed habitats we have tested so far? So this is hermetically sealed lunar Mars habitat that NASA funded to University of North Dakota, where student astronauts can go in and live there for about 30 days without getting any kind of contact to outside. And then successive microbial accumulation can be studied. So we did that, and I'm not going to talk about that today, but if anybody was interested, we published that in Microbiome. So you can find out under inflated lunar Mars habitat. If you type that, you will get that. And then here our fearless Elon Musk in front of the SpaceX, and we did study the this cargo vehicle that transfer our cargo to the space station. So what kind of bugs are there inside that might possibly contaminate the space station? The other one is NEMO project, which is an extreme environment that NASA is working on. It is Aquarius. The name of this particular vehicle is called Aquarius. So 5 to 6 astronauts can go and then train for various days and then see how that is different. Finally, this is called HERA, a Human Exploration Research analog for 500 days in Mars. So we are going to have people, keep it in there for 500 days to see how they can do a lot of things. So all tied up into the closed system. So I will talk about the space station now. So this is the 2D structure of the space station right now, only US side and US partners like Japanese modules are here, and then Columbus module are here. I'm not showing any Russian module here. So inside the Japanese, US module, we have several nodes for several different activities. So these are the nodes that I had a chance to collect. And one thing is, 3 consecutive flights I was able to get. The first and second flight was within 2 months period, but after that the SpaceX 7 blown out, so we were not having any flight to go up to the space, unlike you guys having 1,500 different observations, but we cannot have that much luxury there. So 3 times we were able to get. The third one was after the second flight about a year. Then, so these are the sampling locations. You don't need to read that, but number one is this is the place where astronauts hang around to look at the window to see the Earth from there. So that is called cupola. The other one is, I don't need to explain you what this is, but I don't know why it's not coming. Maybe, okay. So this is nothing but WHC. So where the is space station toilet, and then this is the exercise platform where the astronauts do exercise, and then sweat a lot there. So this is nothing but, anybody guess this? This is dining table. Okay, when they are not dining, they use it as for experiment. So this one is another S5 is another one where the trash and other things are being stored for return back to the Earth. And then this one is a PMM lab, relatively clean. I do not know why. Still, I'm struggling to get the appropriate metadata. Why this S6 is very clean compared to the S5. And then S7 is another a module where some laboratory things are going on. And you see here, there is a little device that's hanging around that is, air filtration also. The squares that you are seeing is the place 1 square meter I was able to collect the samples. Lastly, this is the quarters where the astronauts live in, and then by the side of this, we were able to collect the samples. So what did we do? These are the microbial population parameters we investigated. So we don't want to lose the luxury of these things that you need to have the culture in hand to manipulate a lot of things to get what it does. So we also do culture-based analysis of what is grown in environmental agar medium R2A, and fungus in PDA, and then blood agar pathogens, which is 37 degree. We will incubate this blood agar, but this is 27 degree room temperature. And whatever we are having it, we identified by molecular methods, and also we have the bacteria in hand, so we can do the susceptibility testing with the cultured bugs. And we also do the slew of different methods that are not culture dependent. Main thing is, I keep on asking from the morning, how do you differentiate dead from viable organisms? So there is one key thing that we used, propyodium monoxide technique. I will explain you later. So that PMA treated materials can be used before DNA extraction, and then now you will be able to get the information about the viable microbial burden, the quantitative burden, which is very important. If you are a regulatory agency, you need a quantitative information, otherwise you cannot decide whether your countermeasure, what you are doing is right or not. And then we also do multi-gene metagenomic as well as ITAC sequencing for bacteria, archaea, and fungi. Multi-gene metagenome to know the functional diversity as well as antibiotic resistant genes. This I'll explain you more in detail. And also virulence and pathogenic genes based on this metagenome, and some pathways, unique pathways. And then also this is with the collaboration with Lawrence Livermore and Navy. They have published targeted gene amplification for certain things. So here 500 different antimicrobial genes were targeted directly from the DNA, and then see what kind of genes associated with the antimicrobial resistance are there. So this is a snapshot of culturable bacteria. So this is flight 1, this is 2, this is 3, and then the first one is the blood agar, and then second one is fungi. The third one is I'm showing you is bacterial counts. These are all per meter square. You said low biomass for intensive care units per centimeter square. If you calculate that, this is also equivalent to intensive care unit. But the spacecraft that are built in here before going to Mars is 3 to 4 logs less than intensive care unit. Anyway, so here, in general, all locations yielded more bacteria than fungi. Maybe this is new compared to the mold and other things where things invaded, but keep on hearing the fungus are more than bacteria. But here, space station bacteria are more than fungi. And then obviously some temporal distribution we are seeing. First 2 flights showed same trend, but at least 2 logs more bacteria in third flight. I do not know whether this is a coincidence, because I need to have my other, 4, 5, 6th flight results come in, which is not even happened. So, another 3 years I have to wait. So this is 3 years worth of study anyway. So less than 1 log difference in fungal population among 3 flights. That means it is same. But also spatial distribution we are seeing. So different location yielded more microbes. So 1, 5, and 8 had more microbes compared to the location 6, which I already alluded that has very less bacteria, and also location 3 had less fungi. So what we are seeing? Escape. We are seeing escape here. So, Enterobacter, Acinetobacter, Klebsiella, all these things we are seeing there. These are 8 different points, and then 2 different flights, and some of them are really high in numbers in particular area, but in some places it is there everywhere. This is staph. Coming to fungi, which is Rhodotorula is more and also prevalent in all the places. This is bacterial. This is culturable microbes. Okay. So about 25 whole genome sequence, 25 strains of whole genome sequences are already there. There are biosafety level 2, which is escape. And 25 non-pathogenic strains were also sequenced in collaboration with Jonathan Allen's group, and they are available for further analysis for scientific communities, open right now. So now, if you are interested, you can compare this genome with your indoor genome to see how that compares, whether microgravity is actually influenced. We are already seeing some SNPs and differences are there. So if you are not able to download anything, please contact me. So one of the things that, with the help of Jonathan Allen, we have done this, 21 different strains that are escape organisms, or we call BSL2. So the red one are, present, and the blue one are not present in terms of antibiotic gene patterns. So obviously, some of the things are unique, and that had some, specific genes acquired, and that are equivalent to the erythromycin resistance, which I'm going to tell you later. So I was barking from the morning about culture, whether you can differentiate dead from live with the molecular technology. So which is very important. So PCR is nothing but it amplifies the DNA. I don't need to preach this, people. But the biggest thing is, which is overestimation, right? So we were trying to use the Propiodium monoazide to intercalate the DNA from the dead cells and also naked DNA. Because the Propiodium monoazide is high in molecular weight, bigger, so it will not penetrate through the uncompromised cell. If the cell is compromised, it will penetrate through and block the DNA from amplification in the presence of light. So because of that, the dead DNA and DNA that are naked, as well as the cells that are dead, will not be amplified. So now you have, live cells only targeted. Is that working for everything? There is no one size to fit all. We all know. Around 90% of the things that we tested working. One of the biggest challenge that we have is the spores, which is tough to break open. But spores are less than 1%, right? So at least 99% of the things that you are able to show and reduce the overestimation. So we published in 2013, and ever since we published about 10, 12 papers with PMA. So with that results, from now onwards, I'm showing you only PMA-based analysis, all viable microbes. Okay, this is PMA qPCR for bacteria. This is for fungi, and this is for flight 1, flight 2, flight 3. So what this says, as documented in culture-based methods, the PMA qPCR approach revealed presence of more viable bacteria. If I am doing non-PMA, I am seeing at least 4 to 5 log more than the culture bacteria. Here it's only 1 log more. So, for example, in flight 1, most of them grow. Flight 2, around 10% grew, and flight 3, around 40 to 100% grew. Obviously, there is temporal distribution there. First flight showed 10 to the 6 copy numbers for bacteria, but more CFU. Second and third flight documented with 10 to the 9 copy numbers, where culturable bacteria were around 1 log less. Spatial distribution is also we are seeing, like what we have seen is in culture-based analysis, location 6, we were not able to any PMA qPCR amplification. Obviously, this is a unifrac environmental clustering. Flight 1, flight 2 are different. The control, we have it, which is entirely different. So we are seeing what we are seeing is real. It is not from the control, because one of the locations also. This is location number 3, also falling under the control. So those were maybe the contaminant that we are seeing from the reagents. So based on metagenome, what kind of things we are seeing? Mostly bacteria, less fungi, which we have also seen in the culture-based analysis. And very less virus, which is far on the left, you are not seeing. Some red things are there, and archaea is not even there. So this is based on the family level metagenome thing. So here, as you see, the enterobacteriaceae is more, because you have human living in there, right? So enterobacteria is there, Aspergillosis, all these kind of other family level distributions are there in various things, and also in some other locations that are more different. In some locations is enterobacteria dominant. Okay, so the color different says that red color is flight number 2, blue color is flight number 1, the no color is black color is flight number 3. And also we compared the space station HEPA filter, and also, as I said, space X5 and 6 inside and outside. Before taking to the space station, we also analyzed. So these are the top 10 staph, Methylobacterium, Burgundariales, all these things that I'm mentioning here are the top 10, irrespective of whether flight 1 or 2. Likewise, when you are looking for the fungi, so Penicillium, I'm ruling out the top 2 here, which is even found in the control. So I'm not even taking note of this particular thing. But in general, other than that, Penicillium, Aspergillus, Cryptococcus, and Davidiella are more common, which is also you can see in the regular indoor environment here. So as I said, we are also targeting the amplification of some specific genes. In a paper that Gardner et al. From Lawrence Livermore has done specific for biothreat agent, and in our studies we have concentrated 500 different antimicrobial resistant genes that we know for. So take a DNA that are treated with PMA. Now you hit with 500 in one PCR and then run it by ion torrents to see what you are seeing. So what you are seeing here is this. Even I cannot read this. So I'm giving an example. This particular thing, what you have seen here is for one sample, and then that is nothing but erythromycin resistant gene, MSRB. So this one is from the dining table. So everybody can ask me, is it really coming from the viable cells? Okay, I told you we have that escape organisms, which is BSL level 2. So let us look into the profiles of those cultured bacteria from the same place. So there are 9 different antibiotic resistance that we have tested here. I don't think you can read it, but this one is erythromycin. Okay? So when you see that, as I said, this IFSW4, which is the node 1 dining table, is also being that particular three isolates that I have isolated from there, also resistant to erythromycin. Obviously, this and these are resistant, but I'm concerned right now in this particular profile, one of SW4, where the dining table, the erythromycin, who knows, the astronauts might be taking erythromycin there. And then also the presence of that erythromycin-resistant genes from the Staph aureus culture, ERMA, in this case is MSRB. The other one can be used ERMA amplification. So these were from those isolates 1, 2, 3 that you are seeing it here, 1, 2, 3. These also you are seeing it here. So that actually a confirmatory and validated, technique that we were able to show, in addition to the targeted, amplification, we were able to show that these are present in there. So this morning somebody asked how the environmental microbes are affecting the crew. So that is our next project going on, and then the spring is our flight, and 3 consecutive flight. And in addition to the environment, this is the start. In addition to the environmental surfaces that we did and I showed you, we'll also collect astronaut sampling, crew sampling here, from nasal to buccal to skin and other surface exposed. I'm not doing gut microbiome here. Gut microbiome is being done by some other member of NASA, but I am chartered to do the environmental microbes that's effect on the human and specifically pathogens. So this is coming, and we don't have the results yet. So second one is, we can exploit the environment and then see how the changes are occurring. So one of the things is we deliberately take this Aspergillus, not fumigatus, Aspergillus nidulans and its mutants, and then sending it to the space station to see what kind of secondary metabolites it produces, and then that can be used as a novel drug. So we all know that lavestin is one of the antidepressant compound which can be used for, it is a multi-billion-dollar thing. So to see whether it produces differently under space station. My PI is Clay Wong from University of Southern California on this. The second one, that project is already been sent out, and then we got the sample back and then being analyzed right now. The other one, natural product secretion under microgravity, which is a CASIS project. So this is nothing but Center for Advanced Study in Space Science, which is a nonprofit organization. 50% of the space station real estate and 50% of the crew time are free for anybody who are interested to send anything to space. So if you have a novel idea that you think that space station is a good place to do things, please come and talk to me or send me an email later. We can make your dream comes true, but at the same time, NASA will not pay your research on the ground, but everything will be free. For the flight will be free and bring back to you free. Okay, this one is likewise. We have assembled a team from JPL, Lawrence Berkeley, and then USC and DuPont, to show that we will do some experiment with the already highly known radiation-resistant fungi that has been isolated from Chernobyl. So one thing that is true is under this stress radiation condition, fungi are the first thing popped up, not the bacteria. So that has been shown in this case. The robot went inside, collected the sample in 1986 after the accident. Actually, they sent 2 years after that, and then collected this 28 years of things showing that the fungi are the first one, but they are black mold. In other words, melanin-rich thing. And some interesting properties are this goes towards the radiation source. Towards the radiation source, but not all, but 20% of it. So we selected 1 or 2 from there. We selected some of the previously isolated from space station, 8 of them sent, and then we exposed that to space station. This is ground control, and then brought back after a couple of different time exposure, and then brought back, and then we are doing right now all sorts of omics to see what is different up there, whether it produces any kind of good metabolites that is useful for the humankind. So this is the whole team. Still we are hungry, and all these people are my collaborators. The yellow one are the co-investigators. The postdocs are in this tan color, and the students are in blue color. Thank you very much.